Hello friends, this is the second video of transgenic plants as bioreactors. Okay, how it reacts. Okay, so let's begin. Oleosin partition technology. Let's now start with oleosin partition technology. The purification of foreign proteins through their production in oil bodies is referred to as oleosin partition technology. The oil bodies, along with the oleosin fusion proteins, are quite stable within the seeds and during the course of processing. The pharmaceutical protein of interest remains stable, retaining its biological activity within the seeds for years. Further, the purification oleosin fusion proteins is relatively easier. Hence, oleosin partition technology is preferred for the purification of many pharmaceutically important proteins. Two functional proteins, namely hirudin and beta-glucuronidase, have been recovered by oleosin partition technology. Now, let's know about the production of hirudin in Brassica napus. Hirudin is a natural anticoagulant protein produced in the salivary gland of lychee. It is an effective therapeutic agent as it specifically inhibits thrombin and blocks blood coagulation. Till sometimes ago, hirudin was produced by employing bacteria and yeast. The annual requirement of hirudin is very high and it is a good candidate for producing a transgenic plants. A synthetic gene encoding hirudin was fused with Arabidopsis oleosin gene. An, an, an endopeptidase factor XA gene was inserted between hirudin and oleosin genes. The transgene construct was introduced into Brassica napus. The so developed transgenic plants produce oil bodies containing oleosin hirudin fusion protein. Hirudin can be purified by procedure till now we described. Till now we discussed. Oleosin partition technology holds a great promise for the successful commercial production of several pharmaceutical important proteins. Now let's know about the production of industrial enzymes in plants. There is an ever growing demand for industrial enzymes. Existing sources may not be able to meet the requirements. Large scale production of plant derived transgenic enzymes for commercial and industrial purposes are preferred. Besides several advantages of plants as bioreactors, there is no fear of spread of animal pathogens as is the case with animal production. Let's know about avidin and beta glucuronidase. The first proteins or enzymes that were produced in transgenic plants, that is in maize, are avidin and beta glucuronidase. They are used as diagnostic kits. Now let's know about trypsin. Trypsin is an important proteolytic enzyme and its production by conventional recombinant approaches is rather difficult. Trypsin is now produced in plants. It has a wide range of application for the production of insulin, vaccines, wound care, etc. Phytase. Let's know about phytase. Phytase is a hydrolytic enzyme that catalyzes the hydrolysis of phytate to inositol and inorganic phosphate. For Physate is present in high quantities in many plant seeds used as a feed to pigs and poultry. These animals do not possess the enzyme phytase, hence they cannot derive the nutrient phosphate from phytate. The undigested phytate gets excreted and accumulates in the soil and water, leading to eutrophication. Transgenic plants capable of synthesizing phytase in their seeds have been developed. These seeds can be mixed with the feed of animals and feed. The phytase enzyme has successfully solved nutritional and environmental problems. Now let's know about cellulase and xylanase. The two enzymes, namely cellulase and xylanase, are used in several industries: bioethanol, textile, paper, and pulp. In all these processes, they are basically involved in the degradation of plant materials. By a novel approach, it is possible to produce cellulose and Zealandese in the plants for which the target itself is the digestion of plant cell. The risk of autodigestion of the plant cells is overcome by genetic manipulation to produce thermostable enzymes. Thus, cellulase and xylanase produced by transgenic plants are inactive in the temperatures at which plants normally grow. The activity of these enzymes is restored in on hitting the plant extracts, and these extracts are very uh, successfully used in many industries. While the recovery of enzymes from transgenic plants is often required, there are some instances where crude extraction can be directly used. The industrial application of cellulase, xylanase, and phytase are some good examples of this production. Now let's know about the production of lysosomal enzymes in plants. Lysosomes are the cellular organelles responsible for the degradation of macromolecules. The degradative enzymes present in lysosomes include proteases, nucleases, lipases, glycosidases, phosphatases, phospholipase, and sulfatases. Deficiencies of a specific lysosomal enzyme results in the accumulation of undegraded substrate causing clinical manifestations. Some examples of lysosomal disorders and the corresponding enzyme deficiencies now we'll discuss. First is Tay-Sachs disease. Tay-Sachs disease, that is due to alpha hexosomal alpha hexosaminidase. Gaucher's disease, that is glucocerebrosidase. Horler syndrome, that is iduronidase. Mucopolysaccharidose, that is several enzymes of glycosaminoglycan degradation. Now let's know about glucocerebrosidase. Let's know about glucocerebrosidase. Glucocerebrosidase is a lysosomal enzyme that degrades glucocerebroside to glucose and ceramide. The deficiency of this enzyme causes Gaucher's disease, characterized by swelling of spleen, liver, and bone damage causing extreme pain. Now let's know about Gaucher's disease. Gaucher's disease can be effectively treated by administering the enzyme glucocerebrosidase. At present, the enzyme is obtained from human placenta or from mammalian cell cultures, both being very costly. It is estimated that a single patient requires 10 to 12 tons of human placenta annually, costing 1 lakh crore to 4 lakh 1 lakh dollar to 4 lakh dollar. So it's very costly. Glucocerebroside was the first lysosomal enzyme produced in plants, that is Nicotiana tabacum. The production process of this enzyme has been patented and FDA approved it for use in humans. It is hoped that through the plant-based production of glucocerebrosidase, the patients of Gaucher's disease can be treated in a cost-effective manner. Now let's know about alpha L iduronidase. The deficiency of the enzyme iduronidase causes Hurler's syndrome, the most common disorder of defective mucopolysaccharides degradation. Tobacco plants have been genetically engineered to produce alpha L iduronidase. Now let's know about the limitations for the production of lysosomal enzymes in plants. Both the enzymes referred till now are glycoproteins. Through the series of genetic manipulations, biotechnologists were successful in achieving the desired glycation for the production of glucocerebrosidase and iduronidase. Now let's know about production of antibodies in plants, or directly we can say in short plantibodies. Antibodies or immunoglobulins are the defense proteins produced in mammals. The use of plants for commercial production of antibodies, referred to as antibodies, is a novel approach in biotechnology. The first successful production of a functional antibody, namely a mouse immunoglobulin IgGI in plants, was reported in 1989. This was achieved by developing two transgenic tobacco plants, one synthesizing heavy gamma chain and the other light K chain, and crossing them to generate progeny that can produce assembled functional antibodies. Now let's know about the production of secretory IgA. Secretory IgA is an immunoglobulin that protects against dental carriers produced by Streptococcus mutans. For the production of SIG-A, transgenic plants were developed to synthesize different subunits that then crossed to produce the functional antibody. Four separate transgenic plants synthesizing four distinct pieces of antibody, that is H, L, J chains and secretory component, were developed. The plants expressing H and L genes were crossed, giving plants that produce IgA. When these plants were crossed with plants expressing J chain, the progeny produced dimeric IgA. The dimeric IgA synthesizing plants were then crossed with the plants expressing secretory components. The functional SIGA could be produced. Secretory antibodies have many advantages. They are resistant to proteolytic degradation, hence their yield in plants is substantially higher. SIG-A are the predominant antibodies that protect against mucosal infections of microorganisms. They bind to antigens more effectively and offer good protection. The secretory antibodies produced in transgenic plants have been tested on humans. When SIGA was topically applied to teeth, it could prevent the colonization by Streptococcus mutans up to 4 months. This is comparable to the protection offered by immunoglobulins produced through hybridoma technology. This was possible despite some structural differences between plantibodies and monoclonal antibodies. It appears that there is no differences in the binding properties between the two types of antibodies. Now let's know about the production of other antibodies. Through genetic manipulation, it has now become possible to produce a wide range of antibodies in transgenic plants: whole antibodies, antigen binding fragments, single chain variable fragments antibodies. Some examples of plantibodies and their applications we will now discuss. It is striking to note that despite certain differences in glycosylation of antibodies produced in plants and animals, the plantibodies are by and large comparable in their function with animal antibodies. Let's know about the production of vaccines in plants. Vaccination is an effective approach for proper health care of millions of people in the world. Sometimes the cost of the vaccines is coming in the way, and consequently thousands of children become susceptible to preventable diseases. Vaccines are designated to elicit immune response without causing the disease. In the conventional approach, vaccines composed of killed or attenuated disease-causing organisms are administered. This method is sometimes associated with side effects. With the vaccines, with the advances in molecular biology, recombinant subunit vaccines that are effective in preventing the disease without causing side effects have been identified. There are many production systems for the synthesis of recombinant vaccines. These include mammalian cell cultures, yeast, bacteria, and insect cell cultures, and transgenic animals. Transgenic plants provide an alternative system for the production of recombinant vaccines. The major advantage of vaccine production in plants is the direct use of edible plant tissues for oral administration. By use of edible vaccines or veggie vaccine, the problems associated with purification of vaccines can be totally avoided. The stable or transient expression system can be used to produce vaccines in plants. The procedure adopted for production of vaccine by plant and the use of plants as edible subunit vaccines will discuss. Now let's discuss first about edible vaccine. Plant-based production systems are designed to provide locally available edible vaccines at low cost for easy administration to millions of people. The need for use of edible vaccines arose from the fact that a large number of people in the developing world are the victims of enteric diseases. Edible vaccines provide mucosal immunity against infectious agents. Now let's know about choice of plants for edible vaccines. Most of work on the production of vaccines in plants was initially carried out in tobacco plant that is not edible. These vaccines are now being produced in edible plants such as banana, tomato, and some extent potato. For use in animals, the common fodder crops or food crops can be considered. Some workers consider banana as an ideal system for the production of edible vaccines. This is mainly because it is grown in most parts of the world and eaten raw. Another advantage in that children like eating banana. The procedures adopted for the production of vaccines by plants and use of plants as edible subunit vaccines. Now let's know about delivery of vaccine to the gut. Vaccines being proteins are likely to be degraded in the stomach. This is true to some extent. However, it is now known that the orally administered plant materials can induce immune response. The particulate materials of plants are more effective in this regard. Food-based tablets to replace edible vaccines. Let's know about food-based tablets to replace edible vaccines. It is true that the edible vaccines may work well in the laboratory or under controlled conditions. There is a difficulty of dose adjustment when edible vaccines are consumed as a part of foodstuff. Some workers prefer to discontinue the direct use of plant materials and prefer to opt for food-based tablets containing a known dose of vaccine. This approach is being applied to vaccines produced in tomatoes. Now let's know about the limitations of edible vaccines. Direct consumption of transgenic fruit or vegetable or food-based tablets may create some problems. First limitation is the risk of loss of vaccine by the action of enzymes in stomach and intestine. Second is the possibility of allergic reactions as they enter circulation. Now let's know about the future of edible vaccines. Despite several limitations, biotechnologists see a great hope for the new wave of transgenic veggie vaccines. But it may take several years before the banana or tomato replace the needle as a vehicle for vaccine delivery. Now let's know about the production of therapeutic proteins in plants. The general approaches and the production of industrial enzymes, lysozyme enzymes, antibodies and vaccines in transgenic plants have been described in the next pages. We will discuss. Next slide. Many of these products actually represent therapeutic proteins or biopharmaceuticals. Example, lysosomal antibodies vaccines. Therefore, they must also be referred again. So, the major limitations of protein production by transgenic plants are the low product yield and the difficulties associated with recovery. With improvement in genetic engineering and recovery processes, it is hoped that the production of therapeutic proteins will substantially increase in the coming year. Now let's know about the chloroplast in the production of therapeutic proteins. Chloroplasts are capable of folding the foreign proteins, besides bringing out most of the post translational modification. As the chloroplasts are inherited maternally, they were not spread via pollen to non-transgenic systems. This type of biotechnological approach will help to get clearance from the regulatory bodies of biotechnology. Some of the therapeutic proteins produced in chloroplast now we'll discuss. First is interference, second is serum albumin, third is hemoglobin, fourth is proinsulin, fifth is monoclonal antibody, sixth is protective antigen of bacillus and anthracis. Now completed this part. Any questions regarding this? This part is very vast course. We can't explain this in this small videos. So there are so many things to learn. So to know more about these topics, you can subscribe my channel. Thank you.